Now that we've talked a little bit about how cells divide and that DNA is able to replicate, be transcribed, translated, and produce proteins, we can talk about how these processes give rise to what we call the field of genetics. This involves talking about four major topics. The first one is the basics of Mendel's law. This is probably what you already know about how genetics works. Then we'll talk about variants on Mendel's laws, things that don't fall into two nice categories of recessive or dominant. We'll talk a little bit about the chromosomal basis of inheritance, so we'll tie it back to mitosis and meiosis. And then we'll talk about the difference between sex chromosomes and sex-linked genes. So let's go ahead and start with Mendel's laws. Heredity is the transmission of traits from one generation to the next. Genetics is the scientific study of heredity. So we know that things are inherited from our parents, but through genetics we want to understand why and how. The first person that is largely attributed to being the first geneticist is Gregor Mendel. Mendel's experiments involved crossing pea plants and tracing what he called traits. Traits were things that you could see about the plant, what color it was, how tall it was, and then he would follow those traits from generation to generation. He hypothesized that there were alternate versions of traits, or genes, and he called these alleles. Then that they determined whether or not something was heritable and how. The basic setup of a genetic experiment is as follows. You start with two parents. In this case, we have a purple pea plant and a white pea plant. This is called a parental generation, or filial generation 0. You then cross those individuals together to create progeny. This is the first generation, or the filial generation, F1. In this case, in this version of the experiment, all of the plants in the F1 look like the purple parent. Then you take it one step further. You take two individuals from the first generation and you cross them together. This creates the second generation, or the F2. In this case, we see that 3 of the plants are purple and white has returned. The reason that pea plants were such a good choice is that they are complete flowers. This means that they contain both the female part of the plant, the carpel, and the male part of the plant, the stamen. If you take the stamens and remove them, now the plant can't fertilize itself and you have control over who does the fertilizing. In this case, we have a female purple plant and we're using sperm from the white plant to create the cross. We then get that carpel to mature into peas. And the peas are very easy to plant and observe the offspring. From his research, Mendel came up with 4 hypotheses. We'll describe them below. Your book lists them as very long, but we're going to also give you a shorter version of what they mean. The first one is there are alternate versions of genes. We call these alternate versions alleles, and that they account for variation in inherited characteristics. So for example, you might have a gene that specifies the color of your eyes. Some people have alleles that suggest that their eyes should be blue, and some have alleles that tell their eyes to be brown. The second hypothesis is that for each characteristic, an organism inherits 2 alleles, one from each parent. This makes sense given what we know about meiosis and fertilization. Eggs and sperm are haploid and come together to make a diploid organism. You get one copy of all of your chromosomes from your mother and one copy of all of your chromosomes from your father. This also means that alleles have to have a relationship with one another. For example, an organism that has 2 identical alleles for a gene we call homozygous, and an organism that has 2 different alleles for a gene we call heterozygous. Mendel also came up with a hypothesis for how alleles interact. His idea was that if 2 alleles are different from one another, the allele that is seen is the dominant allele, and the one that is not seen is the recessive allele. To put this into context, if you are homozygous for the blue eye allele, your eyes would be blue. If you are homozygous for the brown allele, your eyes would be brown. When you're heterozygous, which means you have one brown allele and one blue allele, your eyes are brown. We would then say that brown is dominant to blue. Blue is recessive. Sometimes students will conflate the idea of something being common with it being dominant. Whether or not something is common has nothing to do with its relationship to other alleles. For example, let's look at these traits. On the top we see freckles versus no freckles. The dominant trait is freckles. If you're heterozygous, you will have freckles. You have to be homozygous recessive to not have them. But it's very common to not have freckles. Freckles are actually pretty rare. On the flip side, having normal pigmentation versus being an albino has a different relationship. If you're a heterozygous, you will be normally pigmented. If you're homozygous recessive, you could be an albino. Being an albino is extremely rare, so you can see that the thing that is common, no freckles and normal pigmentation, has very little to do with whether or not the trait is dominant or recessive. The last hypothesis was that sperm or eggs carry only one allele for each inherited characteristic, because allele pairs separate or segregate from each other during the production of gametes. This statement is called the law of segregation, and it's related to this idea of independent assortment. So if we were to put this on one slide and make it much shorter, we would say this. Mendel's hypotheses were that alternate versions of genes are called alleles, and they exist. Diploid organisms have two alleles for each trait, one from each parent. If they're the same, they're homozygous. If they're different, they're heterozygous. Alleles that can hide another allele are dominant, and alleles that can be hidden are recessive. And sperm and eggs are haploid, so they only carry one allele for each gene. Now let's apply what we've talked about to the experiment I introduced before. In this experiment, we started with true breeding parents, purple flowers and white flowers. We crossed them together to create an F1 generation where all of the flowers are purple, and at the end, when we cross F1s together, we get an F2 generation where 3/4 of the plants are purple and 1/4 of them are white. So why did this happen? Well, first, every diploid cell has to have pairs of homologous chromosomes. Remember that on those are what we call genes, and genes have a physical location on the chromosome. When we're talking about genes, we'll often assign them a symbol based on whether or not they are dominant or recessive. So for example, we like to use capital letters, like the big B here, to say that this particular allele is dominant to the recessive allele, which we assign a little b. This allows us to come up with what something's genotype is. So what is a genotype? A genotype is the combination of alleles that the organism has. So for example, if we were to look at the P gene here, we could say that there are two big P's. Therefore, the genotype of this organism at this gene is big P, big P. We can do the same thing for the other genes. That means that we have big P, little a, and big B, little b. We would also say that for P, this organism is homozygous dominant, because it has two copies of the dominant allele. It's homozygous recessive for the A gene, because it has two copies of the little a. And it is heterozygous, it has one dominant and one recessive for the B gene. Now we can apply this to our plants. If we cross purple and white together and we get purple, that tells us something about the relationship of these alleles. We talked about how dominant traits can cover up recessive traits, and recessive traits can be hidden. That means that purple is dominant to white. So we can define big P as the dominant allele purple, and we can define little p as the recessive allele white. So if I were to show you a random purple flower, what would you tell me about its genotype? Well, the phenotype of the plant is pretty clear. A phenotype is the appearance of a trait based on its genotype. The phenotype of this plant is purple, so we know that it has to have at least one big P. But big P, little p, the heterozygous state, and big P, the homozygous state, look the same, because big P is dominant to little p. So we don't actually know. We would have to write the genotype as big P dash. What about if I showed you this flower? This is a white flower. Do we know its genotype? Well, we know that the white flower is recessive to purple, and we assigned a little p to the flower. We also know that the only way to be white is to have two copies of the recessive allele, so we do know that this plant has to be little p, little p. Now, I told you in this experiment that we started with true breeding plants. So why do we know that these plants, this purple parent, has to be big P, big P? We know it has to be big P because it's true breeding. I told you, essentially. True breeding is going to mean homozygous. And we know that the little p plant has to be little p because that's the only way to be white. And I also told you it was true breeding. So now we can think about, why are all of the plants purple in the F1? Well, they get one big P from their purple parent and one little p from their white parent. This makes them all heterozygous, and since purple is dominant to white, they're all purple. The F1 generation flowers can give one of the alleles that they have to each of their offspring. So they will give half of their offspring the big P and half of their offspring the little p. Normally when we think about this kind of thing, we can easily put it into what we call a Punnett square. Essentially what a Punnett square does is it takes each possible gametes and puts them on the x and y axis. One axis represents the possible sperm, and the other axis represents the possible eggs. We can then go through and add each of the letters to the box. So for example, all of the top ones will receive a big P from egg, and all the bottom ones will receive a little p from the egg. All the progeny in the first column will get a big P from sperm, and in the second column we'll get a little p from sperm. When we do this, we see that we get the same results that we saw before, 3 purple to 1 white. From a genotypic standpoint, we get the following ratio: 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. This is called a monohybrid cross, and this is the standard genotypic ratio. Our phenotypic ratio is going to be 3 to 1, 3 purples to 1 white. A cross between two individuals that are heterozygous for one characteristic is always called a monohybrid cross, and it's always going to have this same ratio: 3 to 1 for phenotype and 1 to 2 to 1 for genotype. Crosses that involve heterozygotes for multiple traits have different names. The highest number that we'll deal with is going to be a dihybrid cross. In this case, the individuals that are mated in the F1 are going to be heterozygous for 2 things. While you can solve dihybrid crosses as a single event, you can also separate them into the 2 monohybrid crosses that they represent. This is a lot easier when you start doing practice problems, which are going to be very important for understanding this chapter. The reason you can get away with separating dihybrid crosses into the 2 monohybrid crosses they represent is because of the law of independent assortment, which states that alleles of a pair segregate independently from all other allele pairs during gamete formation. So I don't know about you, but I'm tired of using P's, so we're going to use cats. And we're going to use the following cats. We have a solid black cat on the left and a solid gray cat on the right. This difference, whether or not the cat is a dark color or a lighter color, is caused by a gene called dilute. The dominant version of dilute results in a full-colored black cat, but the homozygous recessive individual will be gray. Of course, there are some exceptions like this cutie, but we won't be thinking about them quite yet. So these are trait number 1. The second trait we're going to look at is whether or not the cat has stripes. This is controlled by the agouti gene, which determines the presence or absence of a coat pattern. What exactly those stripes look like is controlled by several other genes, but we won't be thinking about them quite yet. So that you can get an idea of what these traits look like together, the cat on the left would be a black cat that has a pattern, whereas the cat on the right would be a dilute cat that has a pattern. The reason the pattern shows up is because the agouti gene allows for color to be deposited into the hair in a stripe pattern. When the hairs lay down together, they create the illusion of stripes. As you might imagine, having stripes is dominant, and not having stripes is recessive. Now let's set up the experiment. We're going to start with a cat that is homozygous dominant for both traits. That means this cat would be black and have a pattern. We're going to cross that cat to a cat that is homozygous recessive for both traits. That means that this cat will be solid and gray. This is our parental generation. What is the F1 going to look like? Well, the cat on the left can only give dominant traits. and the cat on the right can only give recessive traits. This means that the cat that we get in the F1 is going to have one of each. It's going to be heterozygous for both traits, which means it's going to look like the parent on the left. It will have both a pattern and be black. Under normal circumstances, you should not do this, but we are going to be terrible home breeders. So we are going to interbreed our F1 generation. That means we're going to take two individuals who look the same and are heterozygous for both traits. So we want to ask a couple of questions. How many phenotypes should we get? What's possible? Well, our options are 4. We could get black cats, black tabby cats, which have stripes, dilute cats, or dilute tabby cats. There's a formula that we can use to determine the number of possible phenotypes, and that formula is 2 to the n, where n is the number of genes. We're dealing with 2 genes, so 2 squared is 4. The next thing we can think about is how many genotypes should we get? Each gene has 3 possibilities. For the dilute gene, we could be homozygous dominant, heterozygous, or homozygous recessive. That's what we would expect from a monohybrid cross. And we can also get the same thing for the other gene. We can use this formula 3 to the n because there are 3 possibilities for each hybrid cross, where n is the number of genes. This means that it's possible for us to get 3 squared, or 9 possible genotypes. Now let's go ahead and set up our Punnett square. To do this, we're going to need a lot more squares. So we're going to put the types of gametes that the first cat can make on the top line. Notice that each of these would be haploid. They have one copy of each possible gene. So this cat can give big D big A, big D little d big A, and little d little a. Those are all the possible gametes this cat can make. The other cat is the same, so it's going to put its gametes over here, and we're going to populate that the same way. Big D big A, big D little d big A, little d little a. Now we do the same thing that we did before, and we go ahead and populate each of the boxes with the different gametes from those cats. So for example, in this first box, this cat is going to get a big D and a big A from one parent and a big D and a big A from the other, making it homozygous dominant for everything. So when we fill everyone out, we get this. These would be all of the possible genotypes. Now to make it a little bit easier to see, I'm going to replace these genotypes with what the cats look like. Let's count up the total number of each type of cat. If we think about cats that look like the dominant parent, there are 9 of them. Here they are circled. If we think about cats that look like the black solid phenotype, there are 3 of those. There are also 3 cats that are dilute tabby, and there's 1 cat that's solid dilute. This gives us a 9 to 3 to 3 to 1 phenotypic ratio, and this is the standard ratio for a dihybrid cross. Now the genotypic ratio is a little bit different. So for example, we have 1 that's DDAA, all big, 2 that are 2 big Ds and heterozygous, and 1 that is 2 big Ds and homozygous recessive. This is the same 1 to 2 to 1 ratio as a monohybrid cross, because basically that's what this is. Only one of the traits was heterozygous. Now this is going to keep going. The genotypic ratio for a dihybrid cross is a mess. 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1. I won't be asking you for this, okay? So you don't have to memorize it. But you are going to need to understand where these kinds of numbers come from. This is the point where some students say, but Dr. Sitnik, I hate Punnett squares. Can I just use math? The answer is yes, you can. Unless I explicitly ask you to use a square. What do I mean by that? Essentially, all that we've been doing is thinking about probability. And probability relies on a few different rules. If you understand the probability of something and the rules, you can solve problems like these without using a Punnett square. The first rule is the rule of multiplication. This calculates the probability of 2 independent events both happening. So if we think, for example, about the idea of flipping a coin, we have an individual who is going to be flipping a coin, and that coin's a dime, and another individual who's flipping a coin. That coin is a penny. Now the coins each have 2 outcomes. They can either be heads or tails. This is the same as being heterozygote. You either have the big B, heads, or the little b, tails. No big deal. That means that there's a 50-50 shot that you will flip heads or tails regardless of the corn that you use. If we populate a Punnett square with this, we get the same outcome we would expect before. 2 heads, a heads and a tail, and 2 tails. The likelihood of getting any of these events is the likelihood of each individual event multiplied by the other. So there's a half chance of getting heads on the penny and a half chance of getting heads on the dime. If both things happen, we multiply 1/2 by 1/2, which gives us a quarter. So each of these events has an equal likelihood, and they're each a quarter. Now we add the second rule, the rule of addition, which calculates the probability of events that can occur in multiple ways. So for example, there are 2 ways to get a head and a tail. You could either get a head on the dime and a tail on the penny, or a tail on the dime and a head on the penny. Those are 2 different ways to get the same thing, and so we can add them together. That gives us 2/4. This means that there is a 1/4 chance of getting 2 heads, a 2/4 chance of getting a head and a tail, and a 1/4 chance of getting 2 tails. This is the same 1:2:1 ratio that we see in a standard monohybrid cross. Now that we understand this idea of the rule of multiplication and rule of addition, we can expand this to as many genes as we want without having to make a Punnett square. So let's think about an example. If I gave you a plant whose genotype was big A, big B, little b, big C, and I crossed that plant with a plant that was big A, big B, big C, little c, what is the probability of an offspring having the genotype big A, big B, big C, big C? The easiest way to solve this problem is to treat it like 3 monohybrid crosses. Why don't you give it a try? Alright, so where's the best place to begin? The first thing that you want to do is you want to rewrite the cross. So if we do that, we can see very clearly what we're doing. Big A, big B, big C, cross to big A, big B, big C, little c. We can separate this into the 3 monohybrid crosses. That makes it a little easier for us to look at. And they look like this. And we want to know what is the likelihood that out of these crosses we get this result. Capital letters for everything. To calculate this, we can think about what is the likelihood that each parent will give that letter. So for example, if we're looking at the first cross, the individual on the left can only give big A's. That's all they have. So the likelihood of giving a big A is 100%, or 1. The likelihood of the person on the right giving a big A is 1/2. So the likelihood of getting big A is 1 times 1/2. We can think about this for the subsequent crosses as well. The likelihood of getting a big B from either parent is 1/2, and the likelihood of getting a big C is also going to be 1 times 1/2. That gives us a 1/2 chance of big A, a 1/4 chance of big B, a 1/2 chance of big C. And when we multiply all of these events together, because they're independent events, we get that the likelihood of getting big letters for everything is going to be 1/16. Alright, so now we're going to talk a little bit about special crosses. One of the things that we frequently have to deal with in genetics is unknown. So say for example, you have an individual with a dominant phenotype and you don't know if they're homozygous or heterozygous. There's a special way to solve that. You can do something called a test cross. A test cross is a mating between an individual of unknown genotype and someone who is homozygous recessive for that trait. I gave cats some special treatment earlier, so this time we're going to use dogs. So here are some Labradors. There's a black lab on the left and a brown lab on the right. Black is dominant to brown. Let's say you have an individual dog that is black, and you don't know if your dog is homozygous for the black gene or not. You can breed that dog to a brown dog, and that will tell you whether or not your black dog is homozygous or heterozygous. So how does that work? Well, if your dog is homozygous for the black gene, all of the puppies will be black. But if the dog is heterozygous for the black gene, half of the puppies will be brown. This is because the brown dog can only ever give the recessive trait. So if brown puppies show up, that means that a second recessive trait must have been in the unknown dog. Now let's talk about how we can think about these things for people. A lot of times when we think about genetics for people, we use something called a pedigree. A pedigree is really just a chart that shows how people are related to each other and what generation they're a part of. When we look at a pedigree, there are a few things that tell us information that we need. One is the generation that individuals are part of. So for example, we might consider generation 1 here to be your grandparents, generation 2 your parents, and generation 3 might be you and your cousins. Within each generation, we also number individuals so that it's easy to talk about them. So we might say, we want to talk about individual 3 in generation 3. That would be the filled in black box on the bottom row. In addition to this, circles are going to represent females and squares are going to represent males. And individuals that have the trait are going to be filled in, and individuals that don't will be white. Now we're going to look at a pedigree that's based off of 2 possible traits. One of them is a straight hairline, and the other one is a widow's peak, or the peak that appears above the forehead. The widow's peak is dominant to the straight hairline. So let's look at a pedigree for this family. You can see that the family consists of a lot of individuals that do and do not have the widow's peak. Now we want to answer a question. That question is, what are the possible genotypes for everyone in the family? The easiest way to start thinking about pedigrees is to look for individuals that have to be homozygous recessive, because you know automatically what their genotype has to be. So in this case, that's going to be everyone with straight hairline. All of the people who have straight hairline have to be homozygous recessive. Now we can think of what are the likely genotypes for all the other individuals. If a parent with widow's peak has a child with straight hairline, they must be a carrier for the straight hairline trait. That means that Al, Beth, and Debbie all have to be heterozygous because they all have children that have straight hairlines. That also means that Henry and Isabel in generation 2 must be heterozygous because Kristen, their daughter, has a straight hairline. There are 2 individuals that could possibly be homozygous dominant, but we don't know, and that's Evelyn and Libby. This is because we don't know if Evelyn or Libby are able to have children with straight hairlines, so we can only write their genotype as H-. Now let's try pedigrees from the other side. Let's try drawing one. In generation 1, Frank married to Judy. Both have normal wild-type phenotypes. Evelyn married to John, and both have wild-type phenotypes. This means that we have 2 married couples. We can draw them. Squares represent the boys, Frank and John, and circles represent the girls, Judy and Evelyn. Since none of them have any traits, they would be open or white. And this is generation 1. In generation 2, we're told that Frank and Judy have 3 children, Dottie, Francis, and Eve, and all of them are normal. Evelyn and John have 2 children, Robert and Carrie, and Robert has hemophilia A. So we need to add 3 children for the first couple and 2 children for the second. In this case, one of the children will be filled in. That's Robert. He has hemophilia A. In generation 3, we're told that Eve marries Robert, and they have 2 children, Payne and Kathy, who are normal. So we need to add generation 3, and we need to have them have 2 children who are not filled in because they're normal. Okay, so drawing a pedigree is pretty straightforward. You're just going to draw all of the individuals that are mentioned. So up until this point, we've been working only with this idea of dominant versus recessive, but there are things that fall outside of this very simple framework. So now we're going to talk about variations on Mendel's law. So Mendel's law are valid for all sexually reproducing species, but a lot of time genotype does not dictate phenotype in this simple way. What if, for example, we started with a red flower and a white flower and we crossed them together? Based on our previous understanding of Mendel's law, we would expect all of the flowers to be red, right? Since red is dominant to white here. However, sometimes what happens is instead we get a hybrid or a mixing of the colors. This is a new phenotype, and it's caused by something called incomplete dominance. Incomplete dominance happens when one allele of the dominant gene is not enough to give the dominant phenotype. In this situation, you will end up with 1 red, 2 pink, and 1 white. When incomplete dominance happens, the phenotypic ratio is going to be the same as the genotypic ratio. In this case, that's 1 to 2 to 1. Another time when this kind of Mendelian genetics breaks down is when multiple alleles are expressed at the same time. This is what happens with blood groups, for example. The ABO blood group is actually made up of 3 alleles, not 2, and they are considered co-dominant, meaning that both the A and B alleles are expressed at the same time in heterozygous individuals. This happens because these alleles determine the shape of carbohydrates that are present on the outside of red blood cells. So if you have the A allele, you will have the A carbohydrate. If you have the B allele, you will have the B carbohydrate. And if you have the A and B allele, you will have both. If you're homozygous for the I allele, you'll have neither. These carbohydrates allow your cells to recognize which red blood cells belong to you, and they also dictate which kinds of blood you can receive for transfusion. So for example, people who have type A blood can only receive type A or type O. They cannot have B or AB blood. Type B blood is the opposite. They can receive type B or O, but they cannot receive A. Type AB can receive anything, and type O can only receive type O. So let's think about this in the context of a problem. Maria has type O blood, and her sister has type AB blood. The girls know that both of their maternal grandparents are type A. What are the genotypes of the girls' parents? Let's start by building a very simple pedigree. In this pedigree, we have the girls, their parents, and their grandparents. The first thing we want to do is we want to put on the board everything that we know currently. For example, we know that Maria is type O, and we know that her sister is type AB. We also know that both of their maternal grandparents are type A, and we want to know what their parents are. Now we can think about which genotypes we already know. So for example, we know that the only way to get type O is to have 2 I alleles, and the only way to get type AB is to have 1 A allele and 1 B allele. So we can go ahead and put those on the board. This means that Maria's parents have to have each given her an I allele, and her sister must have gotten an A from one parent and a B from the other. This can only happen if each parent is heterozygote. One of them is AI and one of them is BI, but which one is which? This is where the grandparents come in. We've been told that the maternal grandparents, i.e. the parents of the mother, have type A blood. This means that it's very likely that their daughter is type A. Therefore, we would say that the mother is AI and the father was BI. Up until this point, we've assumed that everything that we see is based off of a single gene, but that's often not the case. Many characteristics result from polygenic inheritance. This is where a single phenotype results from the additive effects of 2 or more genes, and this is very common. An example of that is human skin color. Human skin color is controlled by 3 different genes, in this example A, B, and C. The number of dominant traits that you have for these 3 genes determines the color of your skin. This means that it is highly variable, and in fact you can see that if you looked around your classroom earlier in the semester. This also means that determining where someone lives based on their skin color is actually pretty difficult. For example, if you were to conduct an F1 cross with these 2 individuals that are the same medium shade, you would get all of these possible colors. So just because 2 people are the same color doesn't mean that their kids will be the same color as them. Alright, so now we're going to tie all of this back to the idea of chromosomal inheritance. So the chromosome theory of inheritance states the following things: that genes occupy specific loci or physical positions on chromosomes, and that chromosomes undergo segregation and independent assortment during meiosis. Mendel's laws correlate with chromosome separation in meiosis, and we've already talked about meiosis previously in this class. The idea here is that there is equal probability that chromosomes can line up in one way or another. So for example, when that happens, depending on how chromosomes line up, that's going to change how different alleles end up being inherited, and that this is the basis for things that we see in 9:3 to 3:1. So when we're thinking about what gametes are going to be produced by an individual, we have to consider all possible arrangements. There are, however, of course, examples. Sometimes we don't see a 9:3 to 3:1 ratio, and this happens when genes are linked. Linked genes means that the genes exist on the same chromosome and that they tend to be inherited together. For example, if genes are located on the same chromosome, when meiosis occurs, they will still end up being together. This means that instead of seeing the 9:3 to 3:1 ratio that you would expect from this dihybrid cross, you will instead see a 3:1 ratio, which looks an awful lot like the way a single gene behaves. The exception to this is when crossing over occurs. We talked about crossing over when we discussed meiosis. Crossover events can occur between linked genes, so you can get recombination events, but these are pretty rare. You can calculate this frequency as recombination frequency. I am not going to require you to calculate any of these distances, but I want you to understand what a linked gene is and that it behaves differently than genes that are on opposite chromosomes. We can also use information about recombination rates to tell us where genes are physically located on a map of a chromosome. But for our purposes, we are going to assume that crossing over never happens. If you're interested in recombination frequencies and how crossing over works, I recommend that you take a class on genetics where you will be taught how to calculate this. The last topic that we'll be discussing today will be sex chromosomes and sex-linked genes. Remember how we talked about how there are autosomal genes and there are sex-linked genes? Well, in mammals, the sex chromosomes are X and Y. Other animals use different systems for sex determination, and thus have different types of sex chromosomes. For example, oftentimes in insects, if you have 2 copies of the sex chromosome, you're female. If you have 1 copy, you're male. In chickens and other birds, it's backwards. The homozygotic sex, the one that has 2 copies of the same chromosome, is male, whereas in humans it would be female, and vice versa. And in bees, it's the number of chromosomes. If you're diploid, you're female. If you're haploid, you're male. Now, that's a lot of different things to remember, so for us, we're only going to be doing genetics for things that are like us. A gene that is located on a sex chromosome is called a sex-linked gene. For our purposes, this is mostly going to be genes located on the X chromosome, and that's because the Y chromosome contains very few genes. So let's talk about an example. The gene for this trait is located on the X chromosome. So let's see what happens when we do a cross. The first thing we need to do is we need to talk about the relationship between these genes. So red is dominant to white, and we're going to assign the R allele. Capital R is going to be red, lowercase r is going to be white. Notice that the way I've written it here is as a superscript. This is important. The Y chromosome does not confer anything for this phenotype, so you will be likely to want to leave it out. If you don't put the X there, you might consider that you've forgotten something, or you might forget and treat this like it's autosomal. So please use the superscript. It will help you. In the first version of this cross, we're going to have a female who is homozygous for the red-eyed trait, and a male who has white eyes. Males cannot be homozygous or heterozygous for these traits under normal circumstances. Notice how he only has one X. Whatever X he has is what he's going to look like. Unlike with autosomal traits, for sex-linked genes it matters which parent has the dominant phenotype. So in this example, the female can only give dominant X's to all of her children, and the male can either give a recessive X or a Y. This means that all of the children of this cross will have red eyes, because the female gives an X chromosome to both her daughters and her sons. The females will be heterozygous, and the males will only carry the dominant X and a Y. So when a true breeding female has the dominant phenotype, all of her progeny will look like her. Now let's consider the reciprocal cross. In this situation, the male has the dominant red eye color and the female is true breeding for the recessive color. The female will still give a recessive X to all of her children, but half of the children will receive the dominant X from the male. These will be his daughters, and the Y, these will be his sons. This means that the females will be heterozygous and will have red eyes, but all of the sons will only have the X chromosome from their mother and they will have white eyes. This is a 1-to-1 ratio. That is different from what we would expect to see in a cross for autosomal traits. So when the female is true breeding and has the recessive phenotype, all of the male progeny will look like her and all of the female progeny will look like their father. Most X-linked human disorders are due to recessive alleles and are therefore seen mostly in males. A male that receives a single X-linked recessive allele from his mother will have the disorder. A female has to receive 2 alleles, one from both parents, to be affected. An example for this is something like hemophilia. Hemophilia is an X-linked trait that was relatively common in a lot of royal families. For example, Queen Victoria here was a carrier, and so were her daughter and her granddaughter. Her granddaughter Alexandria was the first to have a male sire, and that was Alexei, and he had hemophilia. There are some basic rules about X-linked recessive traits. They are always passed on from the mother to her sons and daughters, but they are passed from fathers only to their daughters. They are never transmitted from father to son. This is because fathers do not give X chromosomes to any of their sons. Females can be carriers, so these traits are more common to be seen in males, and unaffected females can pass them on to an affected son or an unaffected daughter. Examples of this are going to be things like hemophilia, muscular dystrophy, and color blindness. So if we want to put that as a pedigree, you can see in this particular pedigree that the only individuals that are affected are male. Those would be our solid red boxes. But not all males are affected. For example, if we look at individual 3 in generation 2, we see that none of his children have this disease. If his sons had it, it would be a Y-linked trait. But because they don't, this is an X-linked recessive trait. X-linked dominant traits tend to behave a little differently. An example of that would be Calvin-Lowry syndrome. These are expressed in heterozygous females who can have the disease, and usually no males will have it. This is because dominant traits like this tend to be a lot worse in males, where it is the only X available. So if you see a pedigree where most people who are affected are female, and there are individuals affected in every generation, it's probably dominant and X-linked. Y-linked traits tend to be passed down from males to their sons. This means that every male in the pedigree, if they have a father who is affected, will also be affected. These kinds of traits are relatively rare, but they do occur. This summarizes pretty well the types of inheritance that we've talked about today, with the exception of incomplete and codominance. It's really important that you get experience working on these kinds of problems. Genetics is not the kind of material that you can just memorize. I've included on Canvas some practice problems and a key for answering these problems, as well as an extra credit activity which helps you break down word problems like the kind you might see on your test. Students who do the practice problems tend to do 1/2 to 1 full letter grade better on the final exam than students who don't. If you're still having trouble, please come to office hours and let me know.